All right, and then I would like to welcome you all very much to this ABC seminar. I'm very happy that we could continue doing this, and we make use of the fact that we're doing it online so that we can also invite easily people from very far away. And in that sense, it is a pleasure to introduce José Antonio Gárate to our ABC seminar. He's not a stranger here. He has been in Vienna for a couple of years. First, he actually did his master's degree at the Catholic University in Santiago de Chile. Then his PhD in Dublin with Mile English. And after that, he came to Vienna to join my group for 2.5 years or 3 years, something like that, at BOKU with us. And then in 2013 already, and ages ago, he moved back to Chile with a prestigious homecoming grant from the Fondesit. And currently, since 2016, he's in Valparaiso, I think, and he's currently professor there at the University of Valparaiso at the Interdisciplinary Center for Neuroscience. Still, he's not really going to talk to us about neuroscience, I think, but about graphene. We're very curious what you've done since we've last seen you. And please, the floor is yours.
Okay. Thank you very much, and thank you for attending to this talk. And it's a pleasure for me to kind of go back to Vienna, even though I am far away. But I'm really happy to, because I think this is the third time I give a talk in this ABC seminar. Since I think I did already 2 talks before. So thank you very much. So yes, I'm going to talk to you about a bit, which is a research that actually started with Chris around 2013, 14, and then it has been kind of my side project that started actually before with my PhD, which is about basically studying graphene and carbon nanotubes and small molecules within those particles. So I am not going to talk about neuroscience actually, but this thing might be related. Okay, so first of all, like in my lab, we are very focused on studying what we call nano confinement, which is basically the placement of small particles between nano confined geometries, and that could be carbon nanotubes, for example, which are, I know you could think about tubes that have a diameter of around 4 to 5 angstroms or a bit less. So basically, you have like a single molecule can fit there. And they are quite interesting because basically the thermodynamics is a bit different than from both conditions. We also, of course, given that I am in a group that is doing neuroscience, and my part is basically studying like ion channels, basically, well, we also like to study how this nano-confined process and how this nano-confined water, for example, works in aquaporins with our channeled and transport waters, or other more complex systems, for example, the HV1 proton channel, which we are also studying. So these are the areas that basically my lab is focused on. But now I'm going to talk to you about, and basically we use the nanotubes as a simple model of nano-confinement, and then we try to establish some generalities and understand other more complex processes, such as in aquaporins or the HV1 proton channel. But now I'm going to talk to you a bit more of regarding basically carbon nanotubes and water and graphene interactions. Okay, so this is the main. Sorry, I didn't answer the slides, but basically these are the 2 things I'm going to talk. So for this, let me define like a graphene and carbon nanotube. So basically, probably you all know graphene. Graphene is a single layer of graphite. Actually, it was very nice how it was actually synthesized or found, which is basically by a Scotch tape. They were just doing like a carbon charcoal, and they just do this very nice thing called mechanical exfoliation. That's the technical name, but basically this is the way they found it. And basically a nanotube is a folded. You have graphene and you can fold it. And of course, there are different types of nanotubes depending on how you fold it. So for example, if you follow, this will be like these 2 axes, and if you turn around, so the T will be this will be the folding axis, and then you can define like 2 vectors, and depending of where these 2 vectors are formed, you can form, for example, these armchair nanotubes, which basically they are called armchair because you have this armchair composition at the border here. Then you have the zigzags, yeah, in which you have these two vectors are now have like a 0, 10, for example, zigzag, and you have for example, chirals, which are basically a mixture of these two vectors. Okay, so basically you have these two unit vectors, and depending on the composition of the folding axis, you can define different type of carbon nanotubes. In particular, these different types of tubes have different electronic properties. So for example, the armchairs are metallics, others are semiconductors for all purposes, which is basically fluid or water transport or alcohol transport within them. There have been reported, there are no big differences between these, or at least experimentally, they haven't been, nobody has actually measured differences between, for example, water transport between a zigzag or a chiral or armchair nanotube, okay? Normally, nanotubes are in the ones that we study are in the range of, I don't know, the smallest ones are like 4 angstrom in diameter, and up to 1 to 2 nanometers. This will be called single stamen nanotubes. Bigger diameters you can actually produce, but these are called multiple nanotubes, in which you have actually different concentric nanotubes. Now, why this thing is interesting? Because in the early 2000s, there are like two papers shown by molecular dynamic simulations that actually these carbon nanotubes were spontaneously filled with water. Okay, this was like actually a Chen-Fis letter in 2000, and then Hammer reported this in a very important and seminal Nature paper, in which he actually did study like the thermodynamics of it, and he showed that the filling is spontaneous, and actually water likes to penetrate between carbon nanotubes. So it was very intriguing at that time, because basically graphene and graphite has always been assumed to be hydrophobic materials. So what are the driving forces for this process? And of course, also it gives rise to a very simplistic but experimentally existing model of nano-confined of single-file water, which is present in, for example, aquaporins and other ion channels. Okay, so it was a very nice system to actually study this, what we call one-dimensional systems. Yeah. But the nice thing is what this was also later confirmed by experiments. So, for example, in this paper, by ultracentrifugation, they've actually, with open and closed tubes, they found that tubes that were previously incubated in water, they were actually these bands were lower, and this means that they were being filled with water. Also, the others have shown that there are certain hydrophobic transitions which are dependent on temperature. And finally, they have also been observed. So by electron microscopies, people have observed that these are larger tubes are actually being filled with water. But in this case, in these experiments, they actually did it with a 6 nanotube, with a very small one. So actually, experimentally now we know that nanotubes actually get filled with water. And indeed, and in 2017, if I remember correctly, they were even able to now produce like micelles in which they actually introduce nanotubes within these micelles, and they were able to measure water fluxes within them. So here you see like typical light scattering experiments as function of time from different osmotic gradients, and this for wider and narrow carbon nanotubes, and you see actually that as you increase the osmotic gradient, this curve goes faster into and the light scattering, you know, basically the cell is swollen with water inside. Yeah, you see a change in the light scattering intensity, and this means that the cell the micelle is growing, and you can correlate that with, for example, an osmotic permeability measurement. So this is quite interesting, because now these are being called carbon nanotube porins. Okay, so basically we are being able to produce kind of synthetic channels. okay, so single file water, as I told you, is the placement of water molecules within these nanotubes. You can only fit one water, so here you see in the pore axis here and the radius, and then you see the water, then the oxygen density. So you see that basically one of the key properties of this is that they behave like a kind of dynamic solid. Okay, they are very structured. It's very structured water. And then you see now a top view of the tube. So here you see like the tube, and then you always see that the water is always kind of placed. The oxygen is placed in the center of the tube. So these are the typical characteristics of single file water. This is a 6,6 nanotube. So roughly speaking, it's a nanotube that has a diameter of around 8 angstroms, 0.8 nanometers. So other typical characteristic of this is that, for example, you can study aquí like position correlation. And as you observe this, if you compute this for water within a 6,6 tube, you observe basically that you have a solid-like ordering. Okay, so you don't have like bulk water. Okay, in bulk water you don't observe this. You observe like a single peak, and then it goes flat. And they also produce like a one-dimensional H-bond network. So basically, each water molecule is bonded to the next one. And the loading is very stable. So here you see the amount of water, for example, a 25 nanosecond simulation, and you see that the tube is always filled with water. So these are the typical characteristics. The other one that always people mention is it's basically you have that the waters have what we call a collective dipole. So basically, what you observe is that if you measure the dipole, the alignment of the water dipole with the tube axis, you observe that basically you have two peaks. So in black, you see the average dipole of each molecule, and in red, when you have what we call the collective dipole. So it's the sum of all dipole multiplied by the alignment of this dipole with the z-axis. And of course, you have two peaks. So basically, what you observe is that basically the system points is like an arrow that is pointing up or down. And as you observe here, you can observe the dynamic of this. So in black, we observe the dipole magnitude, the total dipole magnitude of the collective dipole, and then the alignment, and you see that it oscillates between these two conditions all the time. So this is something that, and of course, things that we have done if you apply an electric field aligned with the tube, or if you put, for example, charges that generate a dipole within the tube, you can actually switch this signal to a single position. And of course, this has been related, with the capacity of carbon nanotubes to transport protons, okay? Because given what we call a Grotus mechanism, you can actually, supposedly in these single chains, you can transport proton in a process called proton hopping. Okay, so this was actually reported by De Lago in a very nice review in 2011, if I remember. So where I started to look at this is like, because there is an unusual thermodynamics in non-polar confinement. That's what I like here. For me, the carbon nanotubes are basically a model of non-polar confinement, and this is not only interesting for carbon nanotubes, but also for ion channels, because many ion channels have this kind of hydrophobic cavities. And at that time, this is around 2013, 2012. So I was still working with Chris. There was some controversy whether the pore loading is actually entropically or energetically driven process. Okay, so there were some papers at that time, and for example, in this paper of Hammer on the Heshvaran, they mentioned that actually the process was energetically driven. But then there was a PNA of Todd Pascal that they actually proposed that no, that the process is actually entropically driven, and then they responded again, and they also again said again that no, that they computed the entropy of water within carbon nanotube, and they defined that no, that this is a process that is driven by energy. Okay. So we decided at that time to tackle this issue in a more systematic way. And what we did is basically we did a double decoupling procedure employing thermodynamic integration. So in that case, that we did is like We took a single water molecule, we decouple it from a carbon nanotube, and then we calculate again in solvent. And here you have, like, this is the thermodynamic cycle. Of course, we employed here double decoupling method, so it means that here we apply a restraint, and then we did the corrections from transforming this basically this spring, this harmonic oscillator, into an ideal gas. But of course, we have to add a little correction here, because now we are not dealing with a single binding process. Now you have a binding process that you have a kind of a combinatorial effect, in which you can have, for example, 30 molecules, and you have a combinatorial effect of how do you put 30 molecules from 13 molecules, from 3,000 molecules, for example, of the solvent. So this is a correction that you can look at it in the paper, but we have to add this little correction, which is not very big, but still it's formally correct. And then from these calculations, also this is a very small system, so we're able actually to compute actually the difference in the total energies, and from the difference in the total energies, we were able to compute the entropic contribution. So the idea was to study the whole process, the whole filling process, from an empty tube to a tube completely filled with water, and compute the overall entropic and energetic contributions. So the system that we studied in this case were two periodic tubes, because these systems are very dynamic, so water enters and leaves. So we decided to work with two infinite tubes, or periodic tubes along the core axis, employing the Gromos force field and the Gromos 11 software. So we did the 5.5 and the 6.6. Basically this is one of the narrowest tubes in existence, and it has been reported that in this case water actually doesn't prefer this tube in open simulation. It gets spontaneously filled and emptied, while the 6.6 forms a very stable water loading, and it doesn't get emptied during time. Now, a little trick that we did here is that for the double decoupling method, instead, because we didn't want to interrupt the movement of the water molecules, so instead of applying a restraint to the tube as the particle was being decoupled, we basically did a restraint over a neighbor molecule. So in that way, we were not perturbing the flux along the tube. So here I show you these were a lot of calculations. So we did basically 26 double decoupling calculations. So from the transition from 0 to 13 particles in the 6.6 and 11 for the 5.5. So here I'm showing you the free energy. And as expected, yeah, for the 6.6, you have the free energy. Actually, this is the cumulative free energy. So this is the free energy of adding one molecule, 2, 3, 4, 5, 6. Indeed, you could think that the slope of this curve is kind of a chemical potential. And these are compared with for example, with calculation from the from the spontaneous open tube simulation, in which to basically by the probabilities of the probability distribution, you can also compute the free energies. And the first thing is that we observed that for the 6.6, the open tubes and the closed tubes are quite in agreement until the very last point. And of course, in these points, what we observe is that we have artifacts from the periodicity, because now water molecules start interacting with the other images, and you have deeper energies compared to the ones for the open tubes. But for the 5.5, we observe that the result from just probability distributions are quite different than from our periodic tube, and it means that in this case, periodicity has an important effect. Now, when we look at, we are going to focus on the 6.6, because this is the one that all the other people all these other publications were claiming that this process is actually driven by entropy or by energy. And here I'm showing you that actually it's both. So as you observe, this is the cumulative entropy up to, and ignore this point, because these points, and I told you, these are points in which the tubes are overfilled, and you have star artifact because you have interaction between water between mirror images. So, but the interesting point is that, as you observe, initially the process has an entropic gain. which makes sense because the tube is empty and the water has nothing to interact with. And then at one point, it kind of flattened out. And at these points, then you start observing like a favorable energy. So the process is actually driven by both processes. So what are the driving forces? Why are we gaining energy and entropy, which is kind of counterintuitive? Like we are having entropic gains in confinement, okay? And that's not normally we think that the bigger the volume, the more entropic you are, but in this case, it's like the lower the volume, the more entropic you are. And this is something that it's kind of interesting from these nano-confined configurations. So first, when we look at the energetic components, what we have here is that here I'm showing you in black. Here you're seeing the normalized energy. So it's the energy divided by the amount of water that are within the tube for the electrostatic interaction between the water pair, between the water molecules. In blue, you have the interaction between the waters and the tube and the tube walls. And in red, you have the water with the solvent, which is almost nothing, and in this other black is the van der Waals interaction between the waters within the tube. So as expected, as the molecules are very, very tightly packed, you have a kind of positive value for the van der Waals interaction between the water molecules. It's kind of it's a constant, so it means that the more that a molecule adds, the same contribution it is. Now, in blue, you have the interactions with the pore, which is also is expected that they should be flat, so the more, because they are not dependent on how many molecules you are. So basically, when these are normalized by the amount of water, you have like basically a constant. But the electrostatic interaction between the water molecules, and I mentioned this collective dipole, they are not a constant. Okay, so they change as they increment as I increase the amount of water present in the system. And this, if you look at the previous slide, this will kind of follow this change in the total energy. So the more water molecules I add, the bigger the electrostatic interactions are until you get a certain, and then at the very end they start to flatten out. And this point again, we observe that this is a kind of artifact from periodicity and very overcrowded tubes. For the 5.5, you observe the same, but look at the important thing is that the magnitudes are lower, and actually what is defining why this is more favorable is because the interaction now with the tube it's much lower. It's like 5 or 3 kilojoules lower. So this, I think this is the key factor here, and of course this kind of tight space doesn't allow for the formation of the collective dipole, and this is what is determining that the 6-bit actually prefers it's being loaded on the 5.5 nodes. I must remind you, this is a slight difference. This is only a difference between in the diameter of 2 angstrom or a bit less, or 1.5 angstrom. So it's really these really little details that completely change the thermodynamics. Now, where are these entropic components arising then? Well, one of the things that we look at is the rotational relaxation of the water molecules. So here I show you that from the autocorrelation function of the dipole vector and the HH vector, you can basically fit this to an exponential function, and then you can obtain how much time you can obtain this tau, which is the relaxation time of the process. And quite interestingly, you could think that for bulk water, the HH relaxation time and the dipole relaxation time are kind of similar. So here you have the actual values. It's like 3.20 picoseconds and 2.90. So very isotropic, if you could say. But when, of course, you are inside the tube, you lose that symmetry. So you have that the rotations in this way, like if you have the pore axis, these rotations are very, very, very slow now. So in this case, there are no rotations along the HH vector. But on the other hand, these rotations are quite enhanced. And why is that? Because now the tube actually have the 2 H ones. They don't mind to which partner for the H one to the next water molecule. So they don't care. And basically, you have that the molecule is actually spinning around like this, but not in this direction. So you have a kind of you could compute it, but kind of here you can say that there is an entropic gain in these rotations and an entropic loss in this one. And of course, at the initial stages, because the important here you see that it's kind of flattened out. Now at the initial stages, basically what you have is that basically the water, especially at these stages, is basically entering into almost like a vacuum, and there is basically the water is freely rotating everywhere. So this is where we can explain why at the beginning you have a very entropic gain, you have a very hydrophobic cavity where you have nothing to interact with, and then as the process increases, then you start having more interactions, and then entropy flattens out, and then you have an energetic gain. So at the end, the message for this paper was like, actually the process is driven by both. It's an entropic process and energetic process. And the interesting part is that in confinement, you are not losing entropy, which is kind of counterintuitive. So then later we went to see, okay, let's look at other molecules, because people started to report also that carbon nanotubes, and this is more into some applications, they could serve to actually partition small alcohols like methanol and ethanol. And this is a problem. For example, I come from a country which produce a lot of spirits and wine. And you know that separating methanol from ethanol is quite hard, okay, and it's very expensive. So this could become that if you could actually use nanotubes to actually do this filtering, could be quite cool. And so there were several reports with molecular dynamic simulation, and I think I forgot to put the reference here, but also people have reported that in experiments that carbon nanotubes can partition methanol from solutions. So we also wanted to describe this in a more atomistic and rigorous way. So what we did is like this spontaneous binding experiment simulations, in which basically you put alcohol in the solution, and then basically at different concentrations, and then we look at different tubes. So in black you see like a 6,6, then in red a 6,7,7 and 8. So basically tubes with different diameters. And from methanol and ethanol, and basically what we observe in all simulation that you have a very fast filling kinetics. Okay, so in a way that at relatively low concentrations, like 200 millimolar, you observe that the process, so the removal of water from the tube takes about 15 nanoseconds for a single tube. Okay, so basically this tells us that the process is fast, and that it's kind of, and of course it's dependent on the concentration. In this line, we actually computed the values for a purely diffusive process, and of course these are higher times, because of course you need time, because the tube initially gets filled with water, and then you need to remove the water. So that's why it takes more time than for an empty tube. So it's not a purely diffusive process, but almost. But then we started to look at the configurations of the alcohols, because there is also like an idea that we are trying to push, is that confinement leads to extraction. Okay, and this is something that in biology we know a lot. Like a lot of proteins, for example, they need to be confined by these chaperones to actually fold. Okay, so here we were trying to ask, well, what happens if you put small molecules that are not covalently bonded in confinement? Do they form structure? So here you see like for different pores, which are basically different in their diameters. And the interesting part is that, for example, for methanol in the 6,6, here you have the water molecules, and then you have their dipolar orientations, or their representations as dipoles. And here, as you can observe, you have what we call dipole dimers. So if you see the molecules are forming dimers, that they form dipoles. And the same thing happens, for example, for ethanol in a 7,7 tube. So you have these dipole dimers that you can observe there. Now, for methanol, when you just increase the diameter, you have a transition, and then instead of dimers, you form the collective dipole with some waters inside also, because methanol likes water. So you also have some waters that actually get in between the single phase configuration. So you have what we call dipole dimer to collective dipole transition, which is dependent on the width of the tube. And for from ethanol, it's actually much more visually, it's much more stunning. So you see it here, the dipole dimers, and then you just increase the diameter, and then you have this beautiful chain. Okay. So at that time we were asking, okay, well, we did this with water, so we would like to compute, well, which configuration is more stable, the dipole dimers or the collective dipoles? Which ones are the ones that are more, they have better thermodynamics? But indeed, it's very nice, this nice transition, this nice structural transition, and gives the idea of that confinement kind of leads to correlations, and correlation leads to structure in nano-confined, in very, very small spaces. So we did again the same kind of measurements. So here I'm showing you like the interactions between different partners. So in red you have the interactions of water with the nanotube, not water, sorry, for methanol, with the carbon nanotube. You have methanol with water, because now we also have water inside, so, and as function of the pore load, and methanol interactions. Okay, and if you observe the key parameter, because remember these are dipoles, these for methanol, these are collective dipoles. These are dimers, and this is the collective dipole. And if you look, the key parameter, of course, is that the interaction with the tube here is much lower than in this case. So you have again like 50-50, and here you have 40 kilojoules per mole normalized per molecule. And this wider space, of course, allows to the formation of the collective dipole. And the same thing happens for ethanol. So you see, for ethanol, you have very strong interaction with the tube. Here you have some dimer in the 6-6, in the 7-7, you still have dimers. And then when you reduce interaction with the tube, then you have the nice formation of this collective dipole. And indeed, in this case, water is completely evacuated from the tube. For the case of methanol, you still have some water. As I showed you before, there was still some water between the files. So, and indeed, you can even look at a kind of value. For example, for methanol around 20 kilojoules per. In this point, you have the collective dipole, and it's quite similar for a bit larger, but it's quite similar for ethanol. So these are basically dipole-dipole interactions. Now, this is something that was not very useful, but I like to show, because these systems are so simple that you could actually, sometimes you don't even require to do molecular dynamics to actually simulate them, because basically you can create this kind of very simplistic kinetic models, in which basically what you say is that the transport process is dominated, it's so correlated that as one molecule enters, another leaves. So if you assume that very simplistic model, and then using the fact that Berezovsky developed a model in which you can basically, from the diffusion coefficient within the tube, the actual diffusion coefficient, you can compute the hopping rate, which is the movement, the jumping of one molecule inside the tube and the lid of the tube. Then you can actually get a very simplistic kinetic model. And of course, the K-ion is basically computed from a diffusion limited process. So here you have like basically the capture radius, diffusion constant, and in the bulk, and the water, then the number density of the particles. So with this very simplistic model, you can carry out kinetic Monte Carlo simulations, and you can estimate, for example, survival probabilities, and then you can compare it with simulations. And that's very nice, because in particularly we found, and here I show you like the diffusion coefficients for these different particles in these different conditions. And we observed that the diffusion coefficients for methanol and ethanol were so slow that we were not able, for example, to compute survival probabilities and to obtain proper exponential fits to compute survival probabilities. So, for example, but we could employ these simple models to actually compute survival probabilities for more for longer time. So it is very nice, because here you can use MD to actually parameterize a model and then use kinetic Monte Carlo to actually explore in a more coarse-grained way some kinetic parameters. And indeed, for example, for water, when we compute mean residence times, so basically from survival probabilities, we observe that the values are not that different. But for methane, for example, because we also explore methane here, they are strikingly quite similar. And of course, for methane, this model is almost perfect because basically you don't have the molecules are very simple and you don't have a lot of interactions with the wall. And for methane, it behaves quite well. And indeed, we show that for the other alcohols, this is not good because here we show like the displacement correlation as function of the pore distance. And we said that for water, you have a very good displacement correlation. So it means that water either enters and the other leaves. But for the methanol and ethanol, this is not the case, basically because they really like to interact with the pore wall. But this is it's just nice. I liked to show you that you can actually use this very, very simplistic model to explore the kinetics of this system. But now, then what we wanted to do is the same thing that we did before. So we want to compute the loading free energy of the water loading between the pores. So we employed the same idea, double decoupling method. So we remove the alcohol from the nanotube, and then we appear it and we couple it into the solvent. And we basically have to employ the same thermodynamic cycle as we did before. And of course, initially, I was expecting that, of course, in water, we observed that the the collective dipole, which was forming the 662, was stable, was entropically and energetically favorable. So I was expecting to have the same results for the alcohol. So initially, I expected that this collective dipole, the collective single dipole, was much more stable instead of its free energy when compared to the dipole dimers. So again, now the systems are a bit more complex. So what we have is like methanol at 6,6. So remember, this is dimers, and then we have methanol at 7,7 tube. Then you have a collective dipole, and from ethanol, the 7,7, ethanol at 7,7. It's a dimer, and then it's a collective dipole. And what we did, and we basically wanted to do, is to actually measure the free energy. So basically we did it again, and we disappear the 13, in this case, 8 or 9 molecules, all the loaded particles basically, and via thermodynamic integration. And so here I showed you like all the results. And the most interesting part is that actually we found the contrary. We found that indeed the dipoles dimers are actually more stable than these collective dipoles, which was very strange, and I was not expecting that. So when we look at the curve, so here I'm showing you like the free energy, cumulative free energy for methanol at 7,7. So this is collective dipole, and this is dipole dimers. And then you also observe the entropic and energetic contributions. First, let's look at the 7,7 of the collective dipole, kind of similar to what we found in water. So at the beginning, you will have no big entropic losses, but look at the numbers here are negative. But then, as the collective dipole is being formed, you have a massive loss in entropy. But at the same time, you are forming the collective dipole and you have a massive gain in that. And then from this free energy, basically in this case, we decided you can fit it to a straight line. And then basically the slope of this curve is basically the chemical potential. And this is, we did that because now we have different loads. So the only way we could compare these two results is by comparing the chemical potential, which is basically, as I say, the slope of this curve. But quite interestingly, for the 6-6 system, what we observed is that the entropic values are not as deep at this one. At the beginning, you see that you are not losing a lot of entropy here, and indeed, until the very beginning, just at the very end, you have a kind of a negative entropy. But the important thing is look at this kind of oscillates. So you have like and it's very nice because for uneven numbers, you have a gain in entropy. There's a loss in entropy, and then for even numbers, for uneven numbers, you have the opposite. And it's because when you have one water molecule and it enters, it gains entropy. When you have a pair, it forms the dipole, it forms the dimer, you lose entropy. Then you have a third member, and then you again have more options to form the dipole. So it's very nice that you have this oscillation in the entropic in the entropic gains. And the electrostatic is not as deep as this one, but you also have, you are starting to have like dipole-dipole interactions. And the same thing happens for ethanol. It's exactly the same. So it was very interesting because then we found out that indeed these dimers, by an entropic effect, which is produced by confinement, makes them more stable. So again, you have entropic gains under confinement, which is again very counterintuitive. And here I show you like these results. So in here you have like methane for the dipole dimers. Then you have that the free energy is deeper than for the wider tube. The same for methanol. Is the same thing happens. Not for methane. Methane is another process. For methanol, you have it that the dipole dimers are more favorable than the chemical potential is more favorable than the collected dipoles, and the same for ethanol. Okay, and another thing that we measured in that case is that, well, we say, okay, what happens with graphene? Because graphene is the same material, but it's a different geometry. So now, when you just change the geometry, you basically even change the sign. So there is also a geometric effect here. So this cylinder allows the formation of dipoles, and this actually allows completely change the thermodynamics. So that's also was very interesting, that you have this geometric effect that works, but that is for everything. Okay, so it's for all the molecules, you change the sign. Now, well, this was enough for the small particles. It was very interesting to see this effect of entropic gains under confinement. But then we started to around 2015. I was working in IBM. I did a small scientific stay there, and then we were basically exploring like how proteins interacts with graphene, basically. So here I'm showing you some experiments. In kids here you have atomic form microscopy of different blood proteins, so bovine serum albumin, immunoglobulins, and basically proteins from the blood. And you can observe that basically from these AFM experiments, you can see that actually the proteins absorb onto graphene. And then we also look at circular decreasing, which in that way you can actually check if there are some changes in the secondary structure, and we observe that as function of time. So in black you have the initial circular decreasing profile, and then as you spend 5 minutes or 1 hour, you basically observe that the proteins completely absorb and they basically get unfolded. Okay, and so then we actually studied then the absorption capacity of 3 types of graphene materials. So you have graphene oxide, reduced graphene oxide, and basically carbon nanotubes. Graphene oxide, basically graphene with some OHs, and then reduced. You can, and these processes are done because graphene is very insoluble. So with graphene oxide and other materials, you can solubilize them better. And then you observe that the absorption capacity is quite high. So at the beginning, you observe that the amount of protein absorption, this is measured in milligrams per milligrams of nanoparticle, increases with time. So we also confirming that basically these materials absorb all this type of protein because they're quite specific. And then at that time, I did that. We carried out some simulation, and we are showing you like the interaction energy between the protein and graphene. These are very simple simulations, and we show that basically the protein absorbs via aromatic residues, which is everybody was expecting that aromatic residues, they would like to interact by stacking with graphene. But then we did another cool thing, because basically what we wanted to explore is that it's known that graphene and graphene analysis are quite toxic, and they are quite toxic because basically they are very specific, and they can basically, for example, perturb membranes. So if, for example, you put a graphene layer over a phospholipid layer or phospholipid membrane, the graphene layer will basically start absorbing all the lipids. But simple processes such as pre-incubating the graphene in, for example, blood or bovine serum albumin or any solution that contains protein basically will prevent that effect. So as I say here in simulations, what we did is like we, for example, have a graphene layer, we absorb a protein, and then we check whether how was the absorption capacity of the graphene layer that was now coated with proteins. And as you observe here, when the graphene is coated with BSA, basically you reduce the lipid absorption capacity, which was pretty obvious, but then by experiments, we did the same. And here you see the cell viability. So you have some cell, I don't remember the exact cells, but we have in green, they are alive, and in red, they are dead. And we observe that when you put the pure pristine nanoparticles, they are quite toxic, and particularly carbon nanotubes are much more toxic than graphene oxide. But then if you coat it with proteins, this is called protein corona normally, then basically you can reduce the toxicity, and particularly for graphene. So this, of course, gave us the idea, and this is some unpublished data that we're just working on to, well, because there are many studies of protein absorption on graphene, but not many that have actually focused on interaction between single amino acids and graphene. And I think it's important from a reductionistic point of view to understand what are the driving forces for protein absorption, and for that you need to understand every single residue, basically. So what we did is like we took all 20 amino acids and including their charged and uncharged versions, and we basically did umbrella sampling to actually compute the absorption free energy of all amino acids. So here you see an example that we have, for example, phenylalanine, and we compare, for example, and water. Okay, one would expect that water wouldn't like this is an hydrophobic surface, even though it's not that hydrophobic, at least from the graphene model from in the Gromos force field. But basically, you observe that, for example, phenylalanine really likes graphene. Then here I'm showing you then the absorption free energy of all amino acids reduced by glycine. So basically, we set up the glycine free energy as 0. So then basically here we are looking at the effects of the side chain. But quite interestingly, if you look at it, one would expect, of course, aromatic ones should be the stronger binders. We observe that. But then look at the positive ones, they kind of like it, they like graphene, and in the same range as, for example, apolar ones. And if you look, there is not a clear trend here. But when you plot these absorption free energies as function of the delta Sasa of the surface of the delta surface solvent accessible surface area of the delta Sasa, which basically is a measurement of the interaction surface between the residue and graphene, we observe a very nice correlation. Okay, so basically means that this process is actually driven by the size of the molecule rather than their physical chemical nature, polar, apolar, whatever. And of course here I am cheating a bit because we did this correlation, but we remove the charged residues from this correlation. When you include the charged residue, this correlation is around 0.76. 0.667 or something like that, but there is still a nice correlation. So it was quite, but it makes sense in a way that's why in previous study you observe that basically every amino acid likes to bind to it, and the bigger the protein, the stronger the absorption towards graphene, and it gives us the idea that this process is basically driven by two processes like van der Waals interactions, of course, and water removal. Okay, these are the two driving processes. And so when we look at the other components, because we also we are trying to compute the energy and the energetics and the entropic terms, we observe that for the energy, We also observe like a correlation between all the amino acid, but it's lower. But for the entropic terms, it's kind of lost, and we only observe basically entropic and enthalpic cancellation. So the strong energetic binders are actually also the ones that have biggest losses in entropy. And it is very nice that there is a kind of anisotropy between negative and positive charge residues, which is also very, very interesting. And it's also you can correlate that with other processes, because when you look, for example, at ion channels and voltage sensing, the majority of ion channels contain positive residues within the membrane and not many negative residues. So it seems that there is a thing with positive residues and related to size. Positive residues are much bigger than negative ones when you compare them in weight and size. Of course, so we didn't find a lot of entropic and no correlations with entropy, but with energy. And then let me show you a bit of the examples, the mechanism. So when, for example, for phenylalanine, what you have here is the water-water interaction of the process. So of course, as you absorb the amino acids toward the surface, you have a loss in water-water interactions. Then, of course, the amino acid is also dissolvated. So you see here the amino acid water interactions. But then you have, of course, again, in amino acid graphene interaction, which basically follow a Lennard-Jones. This is just pure Lennard-Jones interactions. And you have also a loss in water graphene interaction. The important thing is that if you look at these are bigger than these. So the dissolution of phenylalanine is not, so the gain in water-water interactions is not compensated by the dissolution of the amino acid. And on the other hand, the gain in the interaction of phenylalanine with graphene is not compensated by the loss in H2 water interactions. And the important thing is that these two processes are basically dependent on the size of the residue. That's all. Like the more water I remove, the more I'm going to gain, and the more water I remove, the more interactions I'm going to gain, and the bigger the amino acid, the stronger the Lennard-Jones interactions are. Now you can look at arginine also, and we have a similar process, but the main difference was for arginine, even though it's absorbed onto the surface, is still kind of solvated, and that was very, 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 very weird. And we are looking at it, but we are observing that arginine, even though we are getting very close to graphene, is not losing a lot of interactions with water, and this makes this big energetic gains, which is not the case for the negative ones. So the negative ones get completely dissolvated when they absorb to graphene. So we are looking at and we are trying to understand that this yet, but these are very, very recent data that I wanted to show you. But then what we did is like, okay, we look at graphene oxide now, because this is normally the material that is being used for applications. And basically when we use graphene oxide, you basically now gain, now you lose this correlation. Now I'm not showing you this here, but now there is no correlation between size and the amino acid, and this is more, and now basically the process is being dominated by basically the residue polarity. And this can be explained because now basically water, you see the absorption free energy is basically within kBT, and basically means that now the free energy preference is all between kBT, because basically the system it doesn't care too much between being in water or being between an oxidized surface. But the other thing that we decided to do, because if you think about it, absorption over graphene is very similar to what we call it's kind of similar to the process of non-polar solvation. For non-polar solvation, many people have been using this method in where they use the delta Sasa or the Sasa of the structure to obtain, for example, the non-polar component of the solvation free energy. So in the same manner, we decided to, okay, let's employ this linear model, this linear correlation, to see if we could predict the absorption free energy, for example, of a pentaalanine over graphene. So here you have that we did a restrained helical pentaalanine and a restrained extended pentaalanine. So these two were restrained because we wanted to avoid of course, when you absorb a helix, it will unfold, but we wanted to basically check if we have particles, if we have a bigger molecule with a different delta Sasa, will our model predict the absorption free energy? And as you observe, and we compare, of course, with the molecular weight, which is of course a very bad predictor because it doesn't involve any type of configuration. And we observe that and for the helical and the stretch, the delta Sasa prediction is quite good. Okay, and particularly for the helical one, we are even within KBT from for rigorous TI umbrella sampling calculation. So it means that this opens the way that probably for hydrophobic or non-polar surfaces, maybe you could, in the same way of methods such as MMA, PVSA, or other methods that compute this kind of non-polar absorption simply by computing the delta Sasas of absorb and absorb state instead of doing the whole process. But of course, we will have to probably we will need more data and test it on other proteins. But it's very exciting that you can actually it works very, very well, and it's very close to the rigorous calculation. But finally, and this is because I'm showing you a lot of this data, and it seems that we have an issue, and it's because in 2018, a group, the group of Pridatko, reported that actually it seems that graphene is not very hydrophobic. So they computed, basically they computed, okay. They computed the contact angle, and basically they showed that the contact angles are below 90. So it means that these things are quite hydrophilic. And this is a very nice method because they basically put a bubble here and they compute the angle in this way. And the nice thing is that we are currently testing basically the use of polarizable graphene models, because normally the majority of graphene models are too hydrophobic, it seems, so that they report contact angles above 90. So in this case, we show you that we are testing polarizable graphene models. And the nice thing is that we observe that when you employ polarizable graphene, so the yellow and red lines here, we observe that we increase the dipole. This is the dipole alignment of the water with graphene. We increase the dipole alignment, and we basically have observed that by including polarizability at graphene, we can actually reduce contact angles. And this actually, we are trying to work now, and we're trying with some collaborators to obtain, to measure contact angles, and in that way actually parameterize a graphene model. So with this, because we are actually finishing, I would like to finish and thank you all for your attention. And these are the acknowledgement of all the funding agencies, and in particular, I would like to acknowledge Mateo, as he has been doing many of the work that I'm showing here. So thank you very much.
Thank you very much, Antonio. Very good, very extensive, very broad topics. Any questions or comments?
So I would have one.
Go ahead, Eka.
Yeah. So you showed at the beginning the carbon nanotubes that you simulated this armchair structure, but did you also consider to simulate the zigzag structures?
Actually, yeah, this is well, normally we started with the armchairs because, well, it was the ones that they were first reported, and the ones that actually, because experimentally it's very hard to determine, if I am correct, you have a different composition of these tubes. What I know is that for transport processes, the zigzags are the armchairs. They don't show any preference, for example, for water transport. That's pretty sure. There are papers showing that. So it seems that these differences in the nanotubes are very important, but for electronic properties, okay, for if they are metallic, semiconductors, and of course here I'm doing just classical molecular dynamics. So these are we are not looking at electrons here, so we don't observe. This shouldn't have a strong effect on the filtering or water loading capacities.
Okay, and then also maybe I missed this, but did you apply some flux on the waters that were going inside of the nanotubes, or they were moving just by thermal fluctuations?
Exactly, this is a spontaneous binding without any driven pressure fluxes or this kind of. Just you put a tube and then it gets spontaneously filled with water. So there are no any pressure, osmotic pressure, or anything like that.
And did you trace some kind of a movement, like they were moving all in one direction, or they were just oscillating? I mean, by looking at your dipoles direction, that you have these double dipoles and suddenly you have this chain and then this circle. Did you notice some movement, like some characteristic movement?
We didn't look at we computed diffusion constant, but this doesn't give you direction, but we didn't, no, there was no preference, flux, or direction. But this is something that maybe is related, because what I wanted to do now, of course, is to look at what happens if I now apply an electric field to the system. I'm going to change this thermodynamic preference towards dipoles or dimers. And this thing, it's maybe like it is interesting because it's also, there has been now reports, for example, in membrane channels, that there is an effect called hydrophobic gating, in which basically they observe that little changes in the diameter that are executed by applying external voltage changes the water concentration in a small cavity. So I think that this kind of little changes produce effects that we can even observe in biology.
All right. Leon?
Hi, José Antonio, nice to see you. So one thing that I'm wondering, I guess you've used reaction field, right, for long range electrostatics. And I'm wondering, in this kind of very specific geometries where every molecule matters and charges matter and so on, do you think there could be some problems and artifacts of special kind introduced by assuming, you know, a homogeneous background a la reaction field? Did you maybe investigate that?
We have looked at, because this I'm here, of course, here I was using Groma, so we use reaction field, but I also have done this with other codes in NMD and with PME, and at least what I can say is that for particle mesh eval, you observe the same thing. So you have the same, at least for water. Okay. I haven't looked at that's a nice question, and maybe I will have to try to look at the how PME will affect these dipole electric formations, but at least for publications and other people using traditional PME methods, they have also observed these dipole dimers and collected dipoles for alcohols.
Okay, and you kind of mentioned in the previous answer, but I'm wondering, are the quantum effects at the boundary really negligible? I mean, you don't have electrons explicitly there, but is there something, because I could imagine, I don't know, right? Individual waters, graphene also having very special, in a way, electrical transport properties and so on. Is there something there where quantum effects could actually be, you know, relevant?
Yeah, it seems that I won't, because these quantum effects normally are a vacuum and a different condition, not in standard biological conditions, the separate conducting properties of graphene that everybody's talking about. But yeah, like the fact that graphene is hydrophilic as I showed you in the last slide, this is changing a lot the concept, because everybody, if you look at contact angles for graphene, in the last, from 20 years, everybody was saying, no, graphene has a contact angle above 90, it's a hydrophobic material. But then lately people have shown that, first of all, graphene has a property that is called transparency. So if you put pristine graphene, basically, normally when you work with graphene, you have to put it over a support like copper or silica or whatever to work on it. And normally what happens is that actually the contact angle is being determined by interactions with the supporting layer and not the graphene itself. And that's what was changing the contact angle. So now that we have this contact angle and going to quantum effects, well, you have a delocalized pi electron cloud. And it's basically can induce polarizability effects, and indeed in a very rough manner, because of course this is not quantum. That's why we wanted to include polarizability in this interaction, because maybe in that way we can capture some of this induced polarizability. But indeed, we also have a colleague working on that. But QM, indeed, if you look at QM and quantum in solvated in water, is very hard to model. Dispersion interactions at the QM level are very hard to model. So this is a real challenge to actually simulate it in conditions that we could apply force fields or models for biology, especially, because now the funny thing still is that this very simplistic model are very good at predicting protein absorption and things like that. Like, so in that case, they correlate quite well with this size dependence with, but still I think we really need, and there is a need to have proper graphite or nanotubes or graphene models that are biocompatible, because now we don't have them.
Thanks.
Dražen?
Hi, José Antonio. Nice to see you again. I'll have a question regarding like the first part of your talk when you were growing in the water molecules in your nanotube and like seeing what is the free energy of filling it up. How, in a way, reliable is the very first water molecule there? Because in principle, you have the periodic nanotube, and then your very first molecule, it actually is basically just looking at the vacuum from both sides. And in principle, when you would actually having it like filled up through like normal diffusion, then on one side you would actually have vacuum, and on the other side you would have bulk. So have you actually tried to somehow mimic this more realistic scenario?
No, actually, that's a good question. No, we haven't, and you're right. Like basically, what you have there is like water in the gas phase, yeah, something like that. But yeah, you could. When you look at the filling, it's so fast that it's actually you will have to kind of moving moving like 3 or 2 molecules at the same time. Maybe that will be more realistic, like to appear 2 or 3 molecules and see if you get the same number. But the important point, and that's why I was not that worried, is that when you look at those values, at least the values that were in between, they were quite in agreement with the open tube simulation. So at least at these intermediate levels, and where you can actually obtain the slope and obtain the chemical potential, you could agree that in that condition it's okay. But I agree that water at the very first water that you're inserting, it is not both conditions, and it's not water with water, and for that reason, it's the same way that we are not looking at actually having a good graphene water interaction, because now I think that thing, even though I forgot to mention, sorry, is that the worst model, not the worst, but SPC with polarizable water. SPC with polarizable water gives the lowest water content. Sorry.
I hope it's not my question.
No, it's not. It's allergies. So sorry for that. But I think that the best model that we have so now that they reproduce this contact angle is actually SPC with polarizable graphene, which is kind of, so I think that probably like we have to start looking at parameterizing graphene in that way, because when you use polarizable water and polarizable graphene, then you have contact angles of around 120. So we really need to look at that, and this is something that it was just reported, and it seems that the traditional, the classical Gromov force field actually gives contact angles with SPC that are not that far from the experiments, but we're still as I said, we are really looking, and we really need to parameterize this stuff.
The magic of SPC, the parameters that Herman Berensen cooked up are just holding up still after 40 years.
Exactly.
Very good. Any further questions or comments? If not, then I will